Hi, my name's Quentin, and I'll be telling you about food timing and circadian rhythms today. The first aspect to talk about is traditional circadian rhythm control, which is surrounding the hypothalamic suprachiasmatic nucleus, which is basically a center in the brain which is entrained to light. So that's sunlight or artificial light, and basically it sends signals through the nervous system and hormones to the body. There's also peripheral oscillators, which are transcriptional oscillators. They are tissue specific, influenced by and influence metabolism, and they indirectly receive timing cues from the central clock via those signals we talked about before. This transcriptional or peripheral oscillator is made up of several genes, some of which to remember are clock, BMAL, PERS, cries, roars, and reverb. And basically, these are a set of genes which regulate each other and basically go through cycles of ebb and flow where some are high and some are low. These peripheral oscillators can be entrained by food timing as well as by food intake and nutrients in general. Glucose triggers repression of BMAL1, acetyl-CoA regulates clock and BMAL1 as well as PER2. So we're basically getting some entrainment of these genetic oscillations based on the consumption of different nutrients. When we don't have nutrients, or when we do, we see changes as well. So NAD plus as well as NADH also regulate BMAL, CERT1, which then regulates clock, and PER2 as well. So we're seeing changes based on what we're consuming and if we're not consuming. You can see here there's very complicated cascades which allow these things to happen. They usually involved energy simulating molecules like CAMP, AMPK, and ATP or AMP. So we're seeing the energy status of the body is really affecting these central clocks as well as the light cycles that we talked about before. Here's some more signals and showing you how they regulate the genetics more directly. So, a good concept to understand here is circadian dysynchrony. Circadian dysynchrony is when there's contrasting peripheral and central rhythms. So, your light cycles and your food cycles are dysynchronized. And basically, you can see changes throughout many tissues in the body, which can be seen in both studies in mice because the light cycles do not synchronize with the food cycles, and we actually see changes going on when you start eating out of alignment with the light cycles of the day. So a study of 10 adults, which was a 10-day study where basically they ate their meals 4 hours later every single day, eventually leading them to be 12 hours desynchronized in alignment, saw that their genetic expression as well as their protein expression of insulin, their glucose levels, and their leptin levels were raised or decreased to levels that represented pre-diabetic or diabetic values in patients. So this can have very profound effects on metabolism. Time restricting feeding is one way to kind of combat this. So that's when you eat in a 4 to 6 hour period and fast for the rest of the day. So that's over 12 hours at least. And what this does is actually allow you to get better oscillation of the genes, which you can see below in those kind of red and green mounds that are represented here. And those are in mice which are either fed in the day, at night, or completely randomly, and when they have light stimulation or not, based on different times of the day. So what is seen here is that when they have light at the same time as they have no food, and when they have food at the same time as they have darkness, since they're nocturnal animals, that they get the best regulation of genes. And when they don't have regulation of when they're eating and they have light all the time, it makes it so that their genetics are completely dysregulated and you don't get any oscillation going on. This time-restricted feeding has been used in resistance-trained males to see the effects. There's 34 of them in this group, and they basically ate within an 8-hour period from 1 p.m. to around 9 p.m. What was seen here is that their body weight was decreased, their fat mass was decreased, leptin increased, so their satiety increased, and inflammatory cytokines went down, including IL-6, TNF alpha, and IL-1 beta. Unfortunately, adiponectin, testosterone, and IGF-1 went down. So in terms of getting muscle growth, this might not necessarily be the very best, but they did maintain their arm and thigh cross-sectional area as well as their maximal strength. What's seen in mice as well is that by consuming your nutrients and fat, particularly in the morning rather than in the evening or their morning, which would be kind of early night versus late, you see much better processing of fats in a lower RER, which basically represents higher fat burning rather than reliance on glucose. So this can be translated into human studies where overweight women were studied for 12 weeks, where they were either fed the majority of their calories in the morning or in the evening for dinner. And what was seen is that those that were fed in the morning had greatly reduced blood triglycerides, total cholesterol, and increased HDL, all of which would decrease the risk of heart disease, diabetes, metabolic disorder, and several other basically metabolic associated and chronic inflammatory disorders. There's also evidence in mice genetics that insulin secretion, protein synthesis, and fat breakdown is optimal when you are consuming those nutrients during typically active phases, light synchronized time periods, etc. So the takeaway from all of this is that time restricted feeding in that first 4 to 6 hours of light and activity is pretty ideal for human metabolic health. A lot of this has been seen in other mammals, but for the most part, you can abstract a lot of this data, and there is human studies as well that represent the fact that time restricted feeding and eating early in the day is a better way to process the nutrients in your body than eating at night and spreading your food intake into dark phases as well as throughout a greater period of time. Thanks for listening.